I'm not muted. I'm going to start out with just a couple of slides, and then we're just going to go through how to sort of create a model in Virtual Cell. So you get used to the interface and some of the decisions that you have to make when you're building a biological, computational model of a particular biology. And luckily, since you're all at home or wherever, you can get up and walk around. So 3 hours straight of lecture is not going to be too daunting for all of you. But anyway, again, sort of feel free to break in at any time that you need. Let's see, I'm just gonna make sure I have all this stuff up. Participant list, okay. And the chat screen. Sorry, it just takes me a minute to create the, where's chat? There's chat. Okay, I have everything up. All right, so this first slide is just to introduce VCell to you again. Again, Les went through the basic equation, which is the kind of problem domain for Virtual Cell. It has a graphical user interface that is designed to allow collaboration between biologists and physical scientists who deal more with the mathematical part of it. And I'm a cell biologist, so I don't go directly to equations. I describe the biology first, and it allows you to enter models from either side, from the math side or from the biology side. As Les talked about, it will handle multiple mechanisms, reactions, diffusion, advection, which is a velocity or flow, membrane transport, and electrophysiology are all handled, and it can do that in multiple geometries: compartmental, spatial, deterministic, or stochastic for all of those, and hybrid solvers as well. We could do some parameter scans and optimization. This, again, is borrowed from Kapazi. But more, Virtual Cell runs on this concept that we have that you're associated with a database. Okay, so as I'll point out as we go through creating models, you log into a database and you save your model to the database for the most part. And you can use our solvers or your local solvers to create simulations. But the association with a model database means that you have access to lots of different models that are already available in VCell and made public or made available to the public or by share. So I'll explain more about that as we go along, but it's important to understand about how Virtual Cell works. So it's not just a standalone program. Also, it has rich annotation methods for annotating the components of your model, so you can remember why you use that particular number for a particular reaction kinetic, for example, and what molecule you are actually talking about when you wrote down C for the molecule instead of actually typing out a name. And it also has a lot of database support for importing models from pathway databases. And it also enables rule-based modeling, which you'll learn more about, I believe, tomorrow. And it can handle a pretty broad range of common problems in biology. In particular, it handles virtual microscopy problems in the sense that because we can model within space, you can have a particular experiment that watches the distribution of fluorescent molecules or changes in ion indicators or probe indicators, and compare the output of a virtual cell simulation directly with your experimental images. And I'll talk more about that as we go along as well. In addition, it can handle ion channels and electrical events, and of course, signaling and metabolic pathways and intracellular trafficking things as well. Okay, so let's go back. It's important to understand the sort of philosophy of building a model within Virtual Cell. So we begin with what we call the physiology, and that is the basic biology. So that's just describing the underlying reactions, the species involved, and where they are located within a cell, in what compartment they are. And that's all done first. And so that creates what we call as a physiology. That includes the kinetics of the reactions, kinetic mechanisms of the reactions, and all of the reactions and where they are. From that physiology, then once that's saved to a database, you can create multiple different applications. So you can do multiple different experiments with that same basic biology. You can use different cell types, for example, different geometries. You can do a deterministic application, or you can do a stochastic application to see if that will change the outcome of the experiments. So you create one physiology and then you create multiple applications from that one physiology. Each application has a unique description of the math. So once you build an application, you create a math description that describes that particular application of the biology in mathematics terms and can be solved in a simulation. So Virtual Cell does that part for you. So it will create the math description from the application. It's also possible to build math models directly from the math as well if you choose, but that will not take you back to an application because that's not a unique flow. The application to the math is unique. So there's a one-to-one correspondence for every application. There's a math description file. From the math description, then you can run multiple different simulations based on different time steps, different simulation specifications, the time step, how long you run it with the mesh sizes, if it's spatial, and you can also do parameter scans and searches. So all of those things are available to you from a single math description file. Okay, so there's many simulations can be run from a particular description. Okay, so that's the overall philosophy of model building within the Virtual Cell software. I also wanted to explain a little bit about the user interface itself before we go there, just to point out that there are, we've broken up the interface into 4 different panes, basically of windows or window panes. So in the upper left, there's what we call the Biomodel Navigator. So that navigates the different parts of the model that you're working on right now. Okay, so we create the physiology and then we have multiple applications of the physiology. So that navigates the different parts of the model you're working on. This part is the main, what I call the main workspace of the model. Its view will depend on what you're working on at the moment, but it's basically where you create the network diagrams and where you can define all the simulation specifications. Below is a selection detail panel, which whatever is selected in the main workspace, the specifics of that will show up in this properties box or pane. And so this is where you add specifics for the kinetics of a reaction, or where you can define annotations, or anything that's specific to whatever's selected at the time will show up in this object properties. And you'll see that as we go along as well. And then finally, on this lower left panel is the database navigator. And so this shows you all of the resources available from databases. Primarily when you log into Virtual Cell, you will find your own biomodels. And this is a picture actually of Les's screen. So those are Les's biomodels. And you'll see also those that are shared with them, public models, biomodels, models that have been made public. So we can do either of those 2 things. You can share with a specific person a particular model, or you can make it so that anybody in the world can see it. And then there's some tutorials that we're going to use today, and some other models that are used for educational purposes. In this picture, we redesigned that a little bit. We also can go to biomodels.net and Pathway Commons. I'll show you those. And you can also search among the biomodels or math models that you have access to. So this window is all of the sort of database accessibility that you need. Okay, so I am going to move on. I see there's one hand raised, Hassan. So do you have a question?
Yeah, can you hear me?
Yes, I can.
Yeah, so I was wondering about the database. Are these database locally hosted, or they are linked with the online repositories?
No, we have a single database that's housed here at the University of Connecticut Health Center. And that's the only central database. You can use virtual cell offline, but you will not then be connected to the database. So there's one central database for virtual cell. Okay, does that answer your question?
Yeah, thanks.
Okay, so I'm going to, let's see, I'm going to get out of my PowerPoint slide and I'm going to go into the virtual cell interface. So I have opened virtual cell and I've connected to the database, and this is what you'll see when you start out. You get a blank window, a blank model workspace, and nothing is open. So the model that I'm going to take you through is actually one of our tutorial models. So if you go to the database window in the lower left, you can open the tutorials, and I'm actually going to work on this one called multi-app, which happens to be the same model that Les used primarily in his discussion of reaction and diffusion. And so it's all the same pathway. And again, I'll just remind all of you that the basic features of this model are that it starts out with a RAN protein, which is basically a nuclear transport protein. When it binds to cargo, which we've called C in this model, then the two form a complex, RAN-C, and that can traverse, flux across the nuclear membrane and accumulate in the nucleus. So, and all of these reactions are going to be reversible, as we'll see. But this is the basic idea of a model. It's a very, very simplified view of a transport between the nucleus and the cytosol. And it's really constructed as a good tutorial. So again, it's not, the real biology is pretty simplified here. Okay, so I'm going to stop at this point. Any other questions? Because I'm going to move out of the tutorial and into a new model so that you can sort of see what you would start with normally. So you're presented with this blank screen and you want to create a model. Okay, the first thing that we're going to do to recreate that tutorial model is we're going to create all of the compartments of the cell. So we're going to define the biology first, and that biology is in the physiology. So we start out at the reaction diagram, and we're going to use this little tool here that looks like a donut for historical reasons. So it doesn't make sense anymore, but it's what we call the structure tool, and we're going to define all of the structure, the compartments in the geometry that we're going to use for this model, right? So it starts out with a single volume compartment, and we're going to click on these lines on the side. They're going to turn green, and we're going to add a membrane. I'm going to do that again, and I'm going to insert another compartment that's going to be the extracellular space. So I'm going to go back to the pointer up here in the tools, and I'm simply going to rename these so we know what they are. That's going to be the extracellular space. This is going to be the plasma membrane. And the cytosol, I can show you, I can actually change the structure name in the properties menu down here below as well. So I'll call this site, okay? So I'm gonna try to line up with the tutorial. We need two more compartments, right? We need the nuclear membrane and the nucleus. So I can, again, add two more compartments over here. I can add a membrane, and I can add another compartment, another volume compartment. And again, we can rename those. This is going to be the nuclear membrane, and this is going to be the nucleus. Okay, so we have all of the compartments that we need to define the biology that we're talking about. So again, the next thing we need to do is make species. So species is these little green balls. Those are the tool we use to create species. So we're going to start out with, I'll make the two species in the cytosol. And again, we always return to the selection tool, and we're going to call this one RAM.
And
yep.
I have a quick question.
Sure.
Can you undo something that you don't want, or if you did something wrong?
You can delete things. You cannot undo. Okay, so I'll point this out as we go along too. Every time I haven't saved anything yet. So if my computer crashes, I've lost everything. So the virtual cell does not save automatically. So what I actually need to do is go and file save as and save the model. Okay, so it's actually in an inconvenient spot, which is down below. I can say, I'm gonna just call this delete multi-app because I don't want to save this one in the end. Okay, and I can save that. So that saves it to the database. Now, if I make a mistake from here on out, I can go, and I'll do this a lot today, I can revert to the saved form. Okay, but there is no undo in virtual cell, and that does become a little bit of a problem in some places. Okay, does that answer your question? Yep.
Yes, it does. Thanks.
I have a question.
Sure.
How did you change the top titles to EC and PM?
Oh, so this little selector tool over on the left here, that arrow.
Yeah.
So if you just select it, you can double click and then type it in there.
And then how do you save it after? Just click enter?
Yeah, then you have to hit enter. Yeah.
Okay, thank you.
Yeah. You can also do it down here in the, here. So I can change it like to lowercase ec down here. Hit enter and it'll change it up there. So either place you can change it.
Okay, thank you.
Yep. And as I'll go in later, you can actually do these in table form as well, but I'll show you that in a minute. Okay, so we have our two species. I'm in the selector tool, so that's going to be C for cargo, right? And then I need to create a reaction between those two to form the complex, right? So the reaction tool is this little line with an arrow on it. And so I'm going to take that and I'm going to move from rand, and it's let it go, it becomes a reaction node, and then I can go further, and that's going to become a product of that reaction. So it created that for me. And then I can connect C to this reaction as well. Okay, so now, oops, sorry, forgot to select the, I did that wrong, I'm sorry. I created another species because I moved it again and created a new reaction. So let me do this again. Ran is gonna go here. And then site C is gonna bind to ran. Okay, and now I'm gonna select the selector tool so I can move these things around. And now I have ran and C form a reaction through a reaction form S2, and S2 I'm gonna say is ran C. Now here, I'm gonna say underscore site. And the reason I'm gonna do that is because this is what's gonna move into the nucleus, okay? But the nucleus is a different compartment. And species are unique to the compartment and must be uniquely named so VCell knows what you're talking about, right? When you create the equation. So I can't have a species named RANC in both the cytosol and the nucleus, okay? So in the flux reaction, it's going to convert RANC in the cytosol to RANC in the nucleus by certain kinetics. So I need the flux reaction now, and that's this little tool here, which is the flux reaction tool. And I can insert that in the membrane. And then I can use my reaction tool to take RANC to the flux reaction and then into the nucleus. It creates a new species for me, and I can rename that species again as, this is going to be RANC and the nuke. Okay. Okay, so now this is the basic biomodel, right? I have RAN combines with C to form RANC in the cytosol. It flows through a flux into the nucleus, which I somehow renamed RAN. Fix that. And it becomes in the nucleus. It becomes located in the nucleus. Okay, so again, this is the basic biology. So the last thing that I have to define is the reaction kinetics, right? So if I select this reaction, which it's called R0, I can name it something. I can name it cargo binding, for example. Cargo binding. VCell cannot handle spaces in any of these names. So if you want a space, you have to use an underscore. And then I need to define the kinetics with which that reaction is going to occur. So the default is a mass action kinetics, where it will put in the forward and reaction rate and the reverse reaction rate. So it's forward rate constant times, in this case, I've drawn the reaction in this direction. That's what those arrows are for. Okay, and so it's forward reaction times RAN times C, and minus the reverse reaction, which is the reverse reaction constant times RAN C site. Okay, it's very straightforward. Now, that's a very simple way to look at it, but I can make it depend on many other things. So for example, I can create another species, I don't know, IP3, let's say, convenient regulator, in the cytosol, and I can make this reaction depend on the concentration of that species. So if I go to that concentration, then I can, there's a number of ways to do this. I can't write in this reaction because I've said it's supposed to be a mass action kinetics, and so that's mass action kinetics. I can change it though to something else. So I can change it to a general reaction. Now, this is a little bug in VCell that will be fixed quickly, but it loses the focus on that properties window. I apologize for that. But now when I do that, you see I've lost my rate expression, right? Because now it's a general reaction, and so it can be, I can type in anything I want here. So I can type in that the forward reaction is the concentration of RAN, oops, sorry, RAN times IP3 times the cargo times some rate constant KR, right? So that could be my reaction. That would be irreversible, of course. So you can write whatever you want there, and now this becomes dependent on IP3, just like it was an enzyme. So I can, currently in release, I can draw a little dotted line from IP3 to the reaction in order to remind myself that IP3 is part of this reaction kinetics. That dotted line does not actively do anything, it's just a reminder. In future versions of VCell, that line will be automatically put in, so you don't have to remember to put it in yourself. Okay, so let's go back to that reaction. Oops. So that's one way I can make this reaction dependent on other things. There's actually an easier way to do it. So, and again, if you see, if you change the kinetics, you lose everything, right? So if I change back to mass action, it goes back to mass action. So if you do that by mistake, it's a good idea to have saved the model already, and then you can revert to saved if you want, oops, I really did want that reaction kinetic. All right, I see I have a few questions here. So I'm gonna stop and answer them. So Amy?
Yeah. So I have a first question. So does this compartmental what you have here, the setup apply for bacteria? Like you want to model the chromosome?
So the compartment can be anything that you want. In general, we consider the there are two types of compartments. There's a volume compartment and there's a membrane or surface compartment, okay?
Yeah.
But you can have two volume compartments next to each other if you want, and then, but you still have to define how things will move from one department to another.
Yeah. So like in your case, you have a nucleus and you also have nucleus membrane. Like
right, you wouldn't do that for a bacterium, right?
right. So I tried this kind of, like I need to set up a compartment like nucleus of for my bacterial chromosome, but then like, so I'm using the spatial model. So in that case, it's automatically generating like, so it's automatically generating like a compartment between the cytosome and nucleus. So that's like when I tried, so I basically like,
It must have a surface. That's the problem. So we can talk later about how to, that's a sort of a little more esoteric of how to create the right boundary conditions between those two compartments. But I'd rather do that. If we could do that offline, that would be better, to be honest.
okay,
It's a little more
sounds
complicated.
good. Yeah.
But there is a way to do it.
Yeah. Also, can I ask, like, what's the differences between different types of the reaction type, like mass action versus like, yeah, I'm still like not.
So these are just different kinetic formulations. So mass action is a forward rate constant times the concentrations of the substrates minus the concentrations of the products times the reverse reaction rate. Okay, so that's a standard form. There's also Michaelis-Menten, for example, which is another standard form with which you need to know Vmax and KMs, okay? General is anything you want it to be. And again, this is more along the lines of what Les was talking about in the first lecture of understanding the different ways of describing reactions based on the knowledge that you have about the reaction mechanism.
Mm-hmm.
Does that help?
So the mass action is recommended for stochastic.
Yes, it's required for stochastic and at this point in VCell.
For non-stochastic, like, but you can also use mass action.
For non-stochastic, sure. Yeah, it's what most people use. It's what we're going to use in this model. Yep.
Yeah, thank you.
Okay, Kamal, did you have a question?
Yes, very quickly. I see that there is a click element called global. You know, in terms
yes,
of.
I will get to that. I'll get to that. Just not
So
quite
the
yet.
other question I have is, how can you split the extracellular matrix in two compartments? Because this is very important when you have the extracellular matrix.
Again, that would be better in an offline conversation. So there are ways of putting two volume compartments next to each other, but it's not common. So it's you have to define the fluxes across whatever the surface is.
Thank you.
Okay. Maybe Masood could talk about that a little tomorrow or Friday as well. Okay, so
I think we should have a geometry mapping little dedicated session for mapping the real geometry surfaces and volumes to actual compartments, because this will answer.
Well, I will do that a little bit, but that's not the same thing as having two volumetric compartments next to each other with no membrane between them, right?
Right, but
That's what they're both asking for.
and you can do that obviously, and
right. So you break in when I get to the mapping part, yeah, and you can point that out, and then we can. I agree, have an offline session for that.
right.
Okay, but just remind me when we get there. Okay, so so that was one way to add sort of a different flavor to this of putting in a reaction, but I'm going to decide I don't want to do that, so I can delete that species, or I can try when I deleted it, because it doesn't depend on this reaction, okay? But let's go back to that reaction one more time and just, if we go back to the mass action, okay, I can make this forward rate constant dependent on, oh, actually I should have left it in there. Let me put that species back. So I can make this forward reaction dependent on that species by simply typing it in here. So I can say that Kf is going to be equal to the IP3 concentration, for example. And silly losing focus. In that case, this IP3 shows up down here as another variable, and the forward reaction, which is uniquely defined for this particular reaction, is equal to the IP3 concentration, which is a species, so it's a variable in the model. I can also make it equal to some other constant that I want to use. So, so now it won't let me delete it because, okay, get it. It won't let me delete it because I have a reaction that depends on it. So it's, VCell is smart enough to say, no, you don't want to do that. So I can turn this back to 0, and then I can delete this IP3. All right, so I can, one more thing with this reaction, for example, I can now make Kf equal to some other parameter. So we'll call it K nu, because I knew I was going to need this. And when I do that, K nu now shows up as another parameter, okay? Now this doesn't have to be specific to this particular reaction, so I can make K nu be important to multiple other reactions. And to do that, I use this global function. So if I click the global, it will go to the bottom. And down at the bottom here, I can make this K nu equal to anything. I can make it equal to IP3 or some value or a function or anything. And because it's global, I can use that parameter in any other reaction I want to use, okay? It's not specific to this reaction, it can be used anywhere. So global parameters are very useful and very important. Okay, so let me get rid of this again. Well, it doesn't matter because I'm going to get rid of this model in a minute. And then one more thing is, again, there's a flux reaction, and here I'm just going to point out that, again, you can use a general flux density. I'm going to go to the tutorial and show you an example of that. I can also use current. It's not used in this tutorial, but we can also include current if we were doing ion channels. And that will include the electrical information here. So I will stop and ask for questions one more time, but this is basically going to be what we call the physiology. It's the structures, the compartments that are involved, the species, and the kinetic form of all the reactions. And that completes basically the physiology. And again, one more thing just before I start, each of these is available in table form. So if you like working in tables rather than with diagrams, you can simply add new reactions this way by specifying where they occur, the name of the reaction, and an equation, A plus B goes to C, for example. And similarly, you can add compartments just by typing them into this structure. And you can add species by typing them into the table. So if you have big models with lots of molecules, sometimes that's easier. All right, I'm going to stop and take some questions now. So Amy, you have a question still? Amy?
Oh, sorry, I didn't.
Okay.
Yeah.
All right, and I'm sorry, I'm not going to be able to pronounce your name from the list, but
Sorry, is Dibujyoti?
Dibujyoti, yeah. So go ahead.
So I have one question. So you just made that case of one reaction is dependent on this species IP3, right?
Uh-huh. Yep.
So instead of a fixed variable, can IP3 be a dynamic entity, like as a part of the reaction?
Yes. Yeah, so it could be the product of some other reaction as well. Absolutely.
So is this a way around of using a mass reaction approach for enzymatic reaction for the stochastic?
Yeah, absolutely. Yeah, it could be you could consider IP3 to be the enzyme in that case, without using Michaelis-Menten kinetics.
Oh, that's
or
great.
You could use Michaelis-Menten kinetics. Yeah, either way you could do it.
Okay.
Yes.
Thank you.
Okay. Just gives you a lot of flexibility. Okay. I'm sorry, I'm going to ruin that name too. Kaustubh?
That's actually pretty close. It's Kaustubh. I just had a quick question. Is it possible for us to create a compartment that is bounded by more than 2 membranes, like 3 membranes or 4 membranes?
No. You can put domains within compartments though. So if you want again, that's a sort of advanced feature with boundary conditions. So if you want one part of the membrane to have different flux conditions than another part, you can do that. And you can specify molecules to be specific to specific regions of the membrane. You can do that as well. But you can't actually have 2 different membranes around one volume compartment, because topologically that just doesn't make sense.
So it would make sense for me to, instead of creating membranes, just create domains to begin with and then map between the domains.
Well, wait till we get to the mapping and the geometry part, and then you'll see how we do it, okay?
Cool. Sorry if I got ahead of myself there.
No, that's fine, that's fine. That's understandable. And then Sarah?
Yeah, hi, Anne. I have a question about the defining rate constant. Like if we have, I don't know, like by mistake, some rate constant like undefined or zero, does it automatically get rid of that rate constant from the reaction
No,
equation?
no, because if the rate constant is zero, then you will have zero flux, right? But it will be there. It's a way of turning
Oh,
off reactions, is setting the rate constant to zero. But
okay,
and sometimes if you add, yeah, no, it won't get rid of anything like that.
okay, thank you.
Yep. And Vlad?
All right. I apologize if I'm getting ahead of time, but is there a way to export this model in SBML format? I poked around.
Yes, there is. There is. So if you go to File, I can just show you that very quickly. So File, Export, and it will give you VCML is our own export, but why does this one not have I don't have, I'm not far enough along to export an SBML model, but when I get to the full model with some simulations, then you can export an SBML model.
You have to create an application.
Right.
In order to export SBML.
But
Okay.
you can
I assume
export
it's going to
that
be
way.
covered, right? Okay.
Yes.
Okay. Thank you.
Yeah, but that's good. That's a good question. Junhao?
Yes, I guess a similar question is because I know that you in VCell use a BioNetGen, then is it able to like import the BioNetGen file?
Yes, it also can import BNGL code.
Yep. Okay.
Import
Pranav?
and export.
Yes. Yeah, Michael will go more over rule-based approaches in the future. Pranathi?
Yeah, hi, Anne. I was wondering if there are a certain amount of species we can have in each section or membrane. Is there like a limited amount or like a maximum or minimum?
No, you can put whatever number you put in there. It might not be appropriate, but there's as far as I know, there's no maximum number of digits.
Okay, and can you put the same species in different compartments, or does it have to?
No, it has to be uniquely named. So there's every species has to have a unique name, and if you used it in one compartment, VCell will not let you use it in another compartment.
Okay, thank you.
So which was why I did this RANSI site and RANSI nuke. It's the same thing, but it's in different compartments. So that was just a convenient way for me to name it. But you must have a unique name.
Okay.
Because otherwise the math doesn't work right.
Okay.
Doesn't know where it is. Okay.
Thank you.
Yep. All right. So I'm going to go on now. Is that everybody's good? I hope. So this is the basic physiology. So I am going to go back now and use not this little toy model that I'm building, but the actual multi-app model. And actually, I'm going to use one that, so I don't mess up the tutorial or saving, I'm going to use my own. So I'm actually going to go back to the model database and search for my model. So I also called it multi-app. So when I search, I find in all the places where the multi-app model is, and I have a lot of them because I keep creating them for tutorials. I'm going to open this one. Okay, so this is now my multi-app tutorial. And the same reaction we saw, and we can see here that here's the flux reaction that's been put in here, as well as a general flux density reaction. So now that I've created the physiology, now I need to create one of the applications. And so what application? I'm going to pick, just to begin with, the non-spatial deterministic, because it's kind of the most straightforward. And then very briefly at the end, I'll have a way to look at the spatial model. Actually, maybe, well, yeah, I'll try and do this really quick. Okay, so, so the spatial model, first thing you come up with is the geometry. So I can also reduce this box if I want to. And so in this case, we're treating each of these compartments as a well-mixed single compartment. And each of those has a size and a volume, a surface area, okay? So in this case, if I put in the size for these, I can change the sizes for these things, and it will give me the relative volume or the surface area for membranes, okay? If you want to know things. We have a little handy calculator here. So if I want a sphere of radius 5, it will give you the volume and surface area to plug into this table. Okay, so the membrane around that volume would be the surface area, and the volume is this. Okay. A handy thing to know, it's very convenient to use an ODE or non-spatial deterministic model to get to a sort of steady state or a reasonable understanding before you go to a full geometry model. But if you create the geometry in the PDE model first, and then you can copy that as a non-spatial deterministic model, it will automatically fill in the right surface area and volume that was found in your geometry. So that's a very useful trick to know if you're modeling. Your ultimate goal is a spatial model, but it's convenient to start with an ODE model because it's much faster. Okay, so that's the structure mapping, and it's all in one compartment, so there's nothing special about the geometry definition. Then you go to specifications, so you just work your way down the tabs in the table in the main workspace. So in this case, we're just going to start with all the RAND-C as a complex in the nucleus. So I've put an initial condition here. This is a really small value, so the stochastic simulation runs fast, so don't worry about it. It's not really very biological at this point. But I can put in any value that I want for these initial conditions for the application. And I can also clamp reactions. That means that clamp species values so it will not change during the reaction. And I can also write rules if that's, again, a little bit more advanced topic I'm not going to talk about now. So the specifications for species. For reactions, all you need to know is you can enable or disable reactions. So if I don't want that to happen in this particular application, I can just not enable that reaction, okay? And fast is if I think the reaction, I want to force it to be considered to be a very fast reaction compared to the other reactions in the model, I can force it to be a fast reaction. Okay, and network relates to rule-based models, so I'm not going to talk about that here. So then I can add some protocols to the model. So protocols can be electrical. So for example, if I'm doing patch clamping or something, I can add voltage changes. If I want just timed events, I can have new timed events and add any one of a number of methodologies for doing timed events. Actually, it's kind of nice. See, I have an event here, but I haven't defined anything. So Virtual Cell provides me with this hint that there's something wrong. That's called a warning. Warnings won't stop me from saving the model, but it will it will be something that might not be mathematically correct, so I should pay attention to it. We also have errors, which is shown down here in the properties, this one particular tab, and let me cancel that so it goes away. So this said no action assigned, it went away because it realized I didn't do anything. It can also, it will show errors. If you have an error here, VCell will not save the model. So VCell will only save things that are mathematically consistent, that it can create math for. And if you do something that it just simply can't figure out how to express that mathematically, VCell will not save your model. So there's a couple of cases where that becomes a problem, and you can always save as a VCML, export as a VCML file, which will save that application as in a standard format or as an SVML file. But better yet, you just go back and fix it. So it's just sort of rare circumstances. But again, it's always good to save your model continuously. And in answer to some of the questions, yes, the videos will be available in YouTube, in that YouTube channel. So you'll be able to see all this again. Okay, so, and then, so those are the protocols, and then finally you get to simulations, and these are the simulations. This little button here is for new simulations, so you can create a new simulation if you want. The simulation now, there's multiple simulations for each application again, and I seem to have lost my, oh, there it is, okay. I'll get my hands back. Okay. I'll answer your questions in just a minute, but let me get through this very quickly. So when I create a simulation, you usually want to edit how the simulation will work. So I can edit the various parts of the simulation itself. I can change any of these expressions, right? So instead of the forward reaction being 1, I can make the forward reaction 10, for example. Or I can scan that. I can create a scan, and I can say take multiple different. I can do it as a list. So I can do 1 and then 10, or I can do a range of values, say from a minimum of 0.001 to 10, and do that in 5 values, and it'll create a simulation that has 5 values for that particular parameter. So these are all the parameters associated with the model, species concentrations, and then I can pick a solver. So VCell usually picks the right solver by default that's best suited for your model. But if you want to force another solver type, you can pick them, and you can also find out a little bit about each solver by clicking the question mark button. But in general, we will, the default solver is usually appropriate. Most of these solvers have automatically determine a time step for you. So you don't need to determine what the time step will be, because it will depend on how rapidly things are changing. So you just put in an ending time. So whether you want a 1-second simulation or a 10-second simulation, you can have a maximum time step. You can change the error tolerances. Again, those are a little bit more advanced features. And then the output options is actually quite important. So you can keep every single time sample, but because the time step is variable, your results will be variable. So you may have a whole lot of results at very short time steps when things are changing rapidly, and then very few. So if you want to compare that to an experiment that has, data every second, then you would want to keep an output interval and just say, I just want to keep every second of this reaction, okay, or every 0.01 seconds. Or you can put in even specific times. Yes, I know, okay. You can put in, specific output times. Oh, this is interesting. Okay. I don't know why I did that, but okay. And then it could put in specific output times. So again, no matter what your output interval is, even if you only save every second, it will still calculate at the appropriate time steps. It just won't save the data out of every single one of those. So the mathematics doesn't change in terms of how the solution is arrived at, only what the data points you're saving is changed, okay? But you might not get an output at exactly the time point you want in your experiment if you don't keep a specific output interval. So you'll play with that a little bit as you get along. Okay. I'm rapidly running out of time here, so I want to get through this pretty quickly. And from here, let me point out that once we've completed the simulation's specifications, then we can run the simulation. And I'm going to go back to this one, this first one here. So there's 2 ways to run simulations. One is I can use this little blue button, and that will run the simulation locally on my own solver, okay? And that runs on my computer, and I get results on my computer, and I can see all of the components of this model in the results menu, okay? So these are just like the images that Les showed, and these are the actual numbers. You can see either one. I can also create an output function here where I can say, have the, site sum, the sum of all the molecules in the cytosol be something like RAND-C site plus C site. And that will create a function so that when I run the simulation, correct simulations. If I run this simulation, then I'll have a new function in the results, which is the sum of those two molecules, okay? So I can use that to create different views of what I'm looking at. All right. So I'm gonna, in the last 5 minutes, I'm gonna talk a bit about spatial finally. I'm glad, I hope all of you have been patient. I'll stop for just 2 quick questions. Junhao, do you have a question?
Yes, specifically for the clamp concentration. So what in what kind of scenario will you use clamp concentration?
There's different scenarios. The one I would use is if something is, I don't want to put in the refreshing reactions, for example, ATP. So I don't want to do the ATP stimulation, as the reactions that stimulate ATP. I just want to have 5 micromolar in my cell and just call it that. I use it for if there's, in a spatial simulation, if I don't want a, if I want to maintain a gradient, or if I don't want the stuff to diffuse away into the ether, I can clamp the solution at like 5 micromolar so it doesn't change around because of diffusion away from the edges of my space, for example. So there's different reasons to do it.
But then in terms of the reaction, then there will be no like a conservation of mass, right?
The confirmation, yeah, it doesn't, there will be conservation of mass, but not for the reaction that created that species. I guess that's the best, it overrides that particular reaction. Is that correct,
Okay.
Jan? Is that the correct way to say it, or less? Anybody listening can help me out here.
It basically says you have an open system now. So there is no longer conservation of mass of that molecule in your system.
Correct.
Because it's now flowing from the outside at an undetermined rate. But the other molecules, they will still be conserved.
Well, let me give you a cute example of where you might want to do a clamped species. So let's say you know that the concentration of calcium inside the cell is 50 nanomolar before or anything else happens, right? Before you have any buffers in the cell that buffer it at that 50 nanomolar. And you know the total amount of buffer, but you don't know the bound form because there are all these different buffers. You may even have an indicator. So you would clamp, you will put that total amount of buffer into the system and put 0 down for the bound species to those buffers, and clamp the calcium at 50 nanomolar, and then run the simulation and let it come to a steady state where you know at that steady state there's going to be the right concentration of bound versus free buffers, because that's where you get your 50 nanomolar calcium, okay? So it's just one example of where you might want to do a clamped species. There are many others. It's a very handy feature.
Okay, if I can get 2 quick questions, because I'm really running out of time. I'm going to snitch about 5 minutes from Pedro's talk. So Gamal, do you have a question?
Yeah, very quickly. Does the software calculate the area under the curve for your simulations instead of giving us the whole?
No, we don't. I don't think we do. But you can easily export that into Excel and do whatever you want with it. Right. So the numbers, you can just copy all the numbers and put them in an Excel spreadsheet. We do have a variety of analysis tools, but I don't think we do area under the curve.
We do allow, if you mean for a spatial simulation, you are able to get the.
Oh, I'll show those. Yeah, for spatial simulations. Yeah.
Thank you.
Okay, and Alma?
Hi. So basically, in your geometry tab, you showed kind of like a 2D diagram of the compartments that you created. So is that the actual connectivity, between them? Can things?
Well, the connectivity between them will be defined by your reactions, basically. So you can actually have something go directly from EC to the cytosol. Now, we do allow that.
Okay, and then the other thing I wanted to ask you about was basically when you're defining, you know, your geometry based upon microscopy images. Like I've seen basically in the PDF version of this tutorial, there's the option to segment manually, but that's actually kind of difficult.
It is.
Yeah, basically, so I tried it, but I couldn't really, like, I kept getting an error that there was something wrong with my geometry, which I figured, you know, is probably just human error. So can people easily just like segment their images using something like MATLAB and then just upload that, you know?
Yes, yes.
Okay.
Yes, you can, if you put in an image with multiple different colors in it, like 5 different colors, one for each region, then VCell will automatically bring those in and then you can map it, in which I'm going to show you actually right now. So
Okay,
good
great.
segue, perfect segue. So Pedro, I'm afraid I'm going to snitch 5 minutes of your time if that's okay. But just to give a brief introduction into the amazing world of space, which where I deal all the time, which is quite complicated. But basically, sorry, let's get the right screen up here. You can create, when you create applications, you create a new application that's deterministic, and then you have to add a geometry to it. And once you add the spatial geometry to it, that'll turn it into a PDE problem. Right, and the way that you add geometries is in what's called geometry definition file, and in this case, this shows you the geometry that I'm using now. This is a slices, sorry, of a data set that we brought in directly from experimental images from the confocal microscope. So, and when you do that, it brings in, we use bioformat, so it'll bring in all the information about the X, Y, and Z spatial scales. If I wanted to replace that geometry, I can do analytic equations in 1, 2, or 3 dimensions, bring in an image from for example, a confocal data set. I can use mesh-based, I can even draw myself on a blank canvas, or I can use constructed solid geometry. So we have a lot of ways to get the geometry in there. Okay, and it has to be segmented, obviously, because we're gonna need to be able to tell virtual cell which pixels belong to which compartment. And that's done in the structure mapping. So another thing you'll find sometimes if you bring in the geometry and you're struggling to segment it and you try to save, it won't let you save the geometry. So that's one of the cases where it gets a little bit annoying, but you have to map the geometry before virtual cell will save it, because otherwise, again, mathematically, it can't figure out what you're trying to do. So you have to tell the software what parts of the geometry belong to each of your structures. So you do that with this line tool, and you just draw a line from the cytosol to whatever in my image is colored brown. Okay, so that segment is brown, and the same for the nucleus, etc. And the structures between the surfaces between those are defined that way as well. So I mapped each of the compartments to the particular structure. And actually, this was when we were talking about doing different boundary conditions. This is the one place that you can do, you can do unusual boundary conditions. Here, you'll see that there's fluxes from one to the other, and you can actually make this a value instead of a flux term, and you can alter the way the compartments interact with each other using these boundary conditions here, but it's pretty rare that you actually have to do that. Okay, so that's the geometry definition. Then when we go to specifications, the one thing that changes here is that now we have a diffusion coefficient. So you need a diffusion coefficient for each of those species. Virtual cell will default to a pretty common protein diffusion coefficient in the cytosol of about 10 micrometers squared per second. I can make that whatever I want. If I have calcium, it'll be more like 300. And if it's a membrane protein, the default, I think, is about 0.1 micrometers squared per second as being a sort of generic membrane protein. And those are usually pretty good because it doesn't change very much with molecular weight. So unless you're talking about something really huge or really small, those values are pretty good approximations usually. Yeah, so, and otherwise it's the same. Here we have one different thing that I'll just point out. And again, if you need to use this, you should talk to one of us. This microscope measurement allows us to collapse multiple species into a single fluorescent species like you would see in a microscope image. So if you have reactions that are involving multiple fluorescent species, you can collapse that all into something we call floor. And that floor will then show up in a final image. And that can be very useful if you're trying to compare it to fluorescent images. And then for the simulations, just the last thing I'm gonna say is, again, when you get to the simulation specifications, now we have this thing called mesh. And here is where you define the mesh size for your reaction. So the mesh size is generally a good idea to maintain a cubic voxel element, especially in 3D, or a square one in 2D. But you can change it if that's better for what you're doing. Again, you can lock the aspect ratio, so if you change any one of these values, it will change the whole thing. And it will show you what the spatial step is now going to be. Often if you save a mesh that's too big, you'll get an error that says you're not representing all of the geometry aspects of your geometry in the final mesh. So it's not a fine enough mesh. We'll let you go back and change it if you want to. We'll also then usually annoy you with a message that says now your simulation is too big, because as you add mesh units, you can see that we get a total size of pretty big. So these simulations can get pretty big pretty quickly. And again, the regular solver is, the default solver is usually the best one to pick. And you have the same choice of different, keeping every time sample or keeping it a particular interval. All right, I'm over time, so I'm gonna stop there and just take one or two more questions, and then we can turn it over to Pedro. So questions, Gamal, do you have a question?
No, you already answered it, sorry about that.
Oh, okay, good, no, that's right. And Amat, did I already answer your question? I guess so. So,
Yes.
Yep, yes? Okay, so anyway, that went pretty quickly, I know, especially at the end, but I'm hoping you get a flavor for how to use virtual cell. The only thing I didn't show you, which I should have, is that if you go to, oh, sorry, I gotta get out of this box. If you go to help and then VCell help, you have a searchable help menu, which we spent a lot of time working on. So you can find a lot of your questions will be answered in the help menu as well. And then of course, go to our tutorials. And a lot of you I'll see in the breakout rooms.